Welcome, everyone, to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled Size Exclusion Chromatography Strategies for the Characterization of Protein Biopharmaceuticals. I'm Jeff Bugaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The complex nature of biotherapeutic molecules often requires an array of analytical techniques for complete characterization. One such technique is size exclusion chromatography, an analytical and purification method that has been used by scientists for decades. While much of the materials that make up a size exclusion column has changed relatively little over the years, detection equipment used to identify eluted compounds has seen a number of advances. Traditionally, size exclusion analysis is performed through the use of UV detectors, which can provide valuable information on protein fragments and aggregates. However, updated detection techniques such as light scattering and mass spectrometry are available to provide more sensitivity and detailed information on eluted products that the modern biopharmaceutical industry demands. Let's meet our panelists for today's webinar who will discuss various protein analysis methods that can be employed when utilizing size exclusion chromatography. Our first speaker today is Maureen Joseph, who is a Biopharma Columns Development Manager for Agilent Technologies. Dr. Joseph will introduce us to some of the principles surrounding traditional aggregation analysis by size exclusion chromatography with UV detection. Dr. Joseph will be followed by Sonia Snyder, an application scientist for Agilent Technologies. Dr. Snyder will discuss alternate size exclusion analysis techniques for protein detection, such as static and dynamic light scattering. Our final speaker will be Kun Sandra, R&D Director of Life Sciences at the Research Institute for Chromatography. Dr. Sandra will provide strategies and data on characterization of antibodies using size exclusion chromatography coupled with mass spectrometry analysis. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply type your question in the Q&A box on the left-hand side of your screen, addressing it to the individual speaker you have a question for, and then hit submit. So without any further ado, let's begin our webinar. Maureen, take it away.
Thank you, Jeff, for that introduction. I'm going to get started today with our presentation on size exclusion chromatography strategies for the characterization of protein biopharmaceuticals. As a general introduction, size exclusion chromatography is particularly important for the characterization of monoclonal antibodies. It's required in order to determine the accurate amount of monomers, dimers, and aggregates present in a particular biotherapeutic. This is necessary because you can have an immunogenic reaction to aggregates, particularly in solution. Size exclusion methods are the first choice for their characterization, and size exclusion can also be used for fragmentation. We'll talk about that on the next slide a little bit. Today we're going to focus on really getting that complete characterization, and in order to do that, we're going to use a range of detection techniques. And this range of detection techniques is going to help improve the characterization. Everybody uses UV for routine monitoring. It's a great choice, does an excellent job. You're testing your final product. You're working in a QA/QC environment. Excellent technique. But light scattering adds a little extra information, and it's very important to get some of that extra information, particularly when you want to know a little more about low concentrations of high molecular weight aggregates. Mass spec is a really powerful technique for size exclusion chromatography. You can get additional molecular weight information on the monomer, dimer, and aggregates, as well as fragments, all at the same time. So let's talk for a second about aggregates. What do we know about them? We know that monoclonal antibodies aggregate. You will have the monomer, dimer, and potentially even higher aggregates. And these aggregates can stimulate an immune response, and that immune response can be mild to severe, so you have the potential for adverse effects. That can impact the safety and the efficacy of a biopharmaceutical drug. Therefore, measuring and understanding aggregation is really important. Aggregation can definitely impact production process economics because you want to keep the amount of that aggregate as low as possible. But you can have manufacturing process steps that increase the level of the aggregates. You can also have stress conditions, heat, the protein concentration itself, any changes in pH, for example. All of these things can impact the level of aggregation. So it's very important to be able to measure it accurately. In fact, it's even required. Regulatory requirements for biotherapeutics require the characterization of aggregates. Here, right out of ICH Q6B, you can see that the category of aggregates includes dimers in higher multiples of the desired product, in other words, the multimers, and they recommend that you use size exclusion chromatography for this. In addition, they talk about looking at multimers and other aggregates through a range of techniques. Well, that's one of the things that we're going to accomplish today. While we don't have a complete range of different techniques, what we have is a range of detectors that really helps us get more information on the monomers, dimers, and aggregates. Before we start looking at all of the data on these, one of the things that the examples that you're going to see all have in common is the use of a BioSEC3 column for aggregate analysis. There are a couple things about this column that are going to help improve our results. It's always the combination of the separation and the detection that gives you the best results. Well, here we're using a 3-micron particle size. 3 micron is easy to use and gives you good resolution. The particular hydrophilic coating on this particle will reduce secondary interactions. Very important when you're trying to understand your results. But in particular, it also helps us because it can be used with low salt buffers. As we start to look at mass spec, we're going to be concerned about the concentration of the buffers in our solution. So this works well for the applications you're going to see today. Here's a very traditional size exclusion chromatography example with UV detection. In this one, we're looking at Herceptin, and we see the monomer peak at 99.6%, and we see the dimer peak at 0.4%. Now, that might be a little bit large for most batches of material, but in this example, we're at 0.4%. What's important is we get the resolution, and we're able to do it, and able to do it routinely by UV detection. Now we're going to start to look at the other detection schemes and what they help you with.
Thanks, Maureen. That was a nice introduction into size exclusion chromatography and a great way to start off our webinar. Before we move on to Sonia's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen, addressing it to the individual speaker that you have a question for, and then hit submit. All of our speakers will be available to answer your questions after the final presentation. Let's start our next presentation. Sonia, the audience is listening.
Thanks, Jeff, for the introduction. Today, I'm going to talk about the characterization of monoclonal antibodies using the Agilent 1260 Infinity Multi-Detector BioSEC solution. Traditionally, aggregation analysis is carried out with size exclusion chromatography. Here, a standard protein mix is injected that contains proteins with different molecular weights, and with the known molecular weight, the column can be calibrated. On this slide, you see here a traditional column calibration indicated by the red line. In size exclusion chromatography, the bigger proteins elute first, followed by the smaller ones. For this classic column calibration, usually a concentration detector is used like refractive index or UV detection. Here I have used the Bio-Rad gel filtration standard for calibration, containing 5 standards. From the very big thyroglobulin with 670 kilodalton down to vitamin B12 with about 1000 dalton. Classic size exclusion chromatography is dependent on the correct eluting behavior of the analyzed proteins. However, if conformational changes occur in the proteins to be analyzed, for example after denaturation or varying protein folding, the elution behavior can change. Post-translational modification like glycosylation, deamidation, or oxidation can lead to differences in the three-dimensional structure of the proteins. Also, proteins can stick to the column due to unspecific hydrophobic interactions. Such sticky proteins elute later than expected and can lead to falsely calculated molecular weights. I will explain this issue later in more detail. So the question is, how can we avoid such false molecular weight calculation? One solution to handle unreliable column calibration is static light scattering detection. With this detection method, the molecular weight is determined without column calibration. On the following slides, I will shortly explain the principle of light scattering analysis. There are different light scattering analysis techniques. In this presentation, I will talk about static and dynamic light scattering. This slide shows the static light scattering principle. In the flow cell of the detector, a laser beam hits the sample molecules and is scattered. The detection of scattering can be measured from several angles. For proteins, 90 degrees gives the best signal-to-noise ratio and is therefore sufficient for an exact calculation of the molecular weight, even for big proteins like thyroglobulin or aggregates of monoclonal antibodies. Using dynamic light scattering, the molecular size of the proteins can be determined as hydrodynamic radius in nanometer. The principle of measurement is completely different from static light scattering. Dynamic light scattering measures the Brownian motion. The Brownian motion is the thermodynamic motion of particles in liquids or gases. Depending on the size, the molecule moves faster or slower. Usually smaller molecules move substantially faster than bigger molecules. This motion is monitored, and using a correlation function, the hydrodynamic radius can be calculated in nanometer. Here we see examples for the relatively narrow range of hydrodynamic radius values for proteins. It is typically in the range of about 1.5 to 10 nanometers. The molecular weight changes more rapidly than the hydrodynamic radius as the size of the molecule increases. On this slide, you see the Agilent system solution for the analysis of protein aggregates. On the left, the Agilent 1260 BioInert LC, consisting of a BioInert quaternary pump, a BioInert autosampler, a column heater, and that followed a DAD with BioInert flow cell. After DAD detection, the sample reaches the light scattering detector, a combination of static and dynamic light scattering detection. With this setup, the complete flow path is BioInert and has a high salt tolerance for the most salty SEC buffers. The light scattering detector is housed in the multi-detector suite, enabling additional detection options, for example, RI or viscometry detection, as there is space for 2 detectors in the multi-detector suite. One of the big advantages of light scattering detection compared to UV or RI detection is the high sensitivity to high molecular weight material, even if the aggregates are very low concentrated. This fact allows very sensitive detection of aggregates and oligomers. This slide shows an overlay of RI signal and light scattering signal of a protein monomer and some aggregates. Especially the higher molecular weight aggregates are only visible in the light scattering signal. On this next slide, you see a case study of a monoclonal antibody which was heated to 60 degrees for 1 hour to generate a small amount of aggregates. The black signal is the UV signal at 280 nanometers. The red signal is the light scattering signal at 90 degrees. With this slide, I would like to emphasize the high sensitivity of light scattering detection versus UV detection for aggregates, with a clear peak in the red signal trace compared to a very small peak in the black UV signal. Here you see the analysis of thyroglobulin. The blue lines represent the molecular weight of thyroglobulin monomer and the different thyroglobulin aggregates. In the table below, you see the determination of molecular weights of the monomer and the aggregates. Peak 4 is the monomer. Peak 3 has about the double molecular weight and can therefore be considered as the dimer. Looking at the molecular weight of the second peak represents the tetramer, and the first peak, the 10-mer of the tetramer, so a very high molecular weight multimer. In addition, the combination of the DAD as a concentration detector with the light scattering detector, a determination of the relative aggregate concentration becomes possible. Now, I would like to take a closer look at the advantages of the light scattering detection. On this slide, you see the analysis of a monoclonal antibody. On the left, the UV detection in combination with a column calibration as the classical SEC analysis. The molecular weight was determined with this method as resulted at about 150 kilodalton. On the right, you see the analysis of the same antibody with static light scattering at 90 degrees. The molecular weight was also determined at 150 kilodalton. For this antibody, the molecular weight determination is absolutely comparable with both detection methods. But what happens if the antibody does not elute like we expect? This slide shows you an example. Again, on the left side, you see the classical column calibration with UV signal. 3 to 4 peaks are visible. For 3 peaks, the molecular weight was determined using the calibration curve. Following molecular weights were determined: 145 kilodalton for the first peak, 1 kilodalton for the second, and 100 dalton for the third peak. Analyzing this sample with light scattering detection on the right side, you get absolutely completely different results. With light scattering, every peak was determined at about 150 kilodalton. So consequently, I came to the conclusion that all peaks represent the same antibody, but only small peaks of it elute properly, and most of the antibody sticks to the column. The result was confirmed with dynamic light scattering analysis with a molecular size of about 5 nanometers as hydrodynamic radius, which is often found for antibodies in literature. The next slides show the excellent reproducibility of our system. Here we see the overlay of 7 subsequent injections of a pharmaceutically used antibody. The precisions of retention time and area, as well as the precision of the bulk molecular weight and the hydrodynamic radius, were excellent. Here we see an overview of the molecular weight determination together with the related relative standard deviations for 5 different proteins from cytochrome C up to thyroglobulin. Different amounts of each protein were injected and the molecular weight was determined. The relative standard deviations were calculated over several injections with different injected amounts. The relative standard deviations did not exceed 3%, not even for very big proteins like thyroglobulin, nor for small proteins like cytochrome C with 11 kilodalton. Depending on the sensitivity of the 90-degree static light scattering detector, the injection range was adjusted for every protein. The sensitivity of the light scattering detector increases with the size of the protein. Except for thyroglobulin, all molecular weight determinations agreed well with the literature values. For thyroglobulin, the deviation was higher due to insufficient resolution of the thyroglobulin monomer from its aggregates. The better the resolution within a chromatogram, the more exact is the molecular weight determination as observed for ovalbumin. For those of you who want to know more about the Agilent 1260 Bio-SEC solution, there are several application notes found on the Agilent webpage.
That was great. Thanks very much, Sonia. I think our audience now has a great sense of the power and versatility of light scattering analysis in combination with size exclusion chromatography. It looks like we're getting some really nice questions already, and I want to encourage the audience to keep them coming. Let's begin our final presentation for this webinar. Koen, when you're ready, you have our attention.
Okay, thank you, Jeff. So after this nice overview of the multi-detector by Sonia, I will switch gears now a little bit and discuss on yet another detector that can be combined with size exclusion chromatography, and this is of course mass spectrometry. Now, as has just been demonstrated, size exclusion chromatography is really very valuable to assess aggregation and also fragmentation, and these are really considered critical quality attributes of monoclonal antibodies and also other protein therapeutics, and these might really impact on product safety and efficacy. Now, with the detection techniques just described, the identity of the observed peaks can typically not be obtained. Of course, fractions can be collected and further analyzed using mass spectrometry, this typically after desalting, but this is a process that is rather time-consuming. Now, imagine, however, that size exclusion and mass spectrometry can be combined in an online fashion. This would really open up new possibilities towards the detailed characterization of monoclonal antibodies. Now, size exclusion chromatography typically makes use of buffers containing non-volatile salts, such as a phosphate buffer. Now, unfortunately, these conditions are not compatible with mass spectrometry. Now, on this slide, the upper UV chromatogram shows the separation of the monoclonal antibody Herceptin on the Bio-SEC column using a 150 millimolar phosphate buffer at pH 7. Now, under these conditions, sharp peaks are obtained and aggregates and fragments can be detected at levels as low as 0.1% using UV detection. Now, spraying a phosphate buffer in the mass spectrometer, however, would give rise to a dirty mass spectrometric source, and moreover, we would not observe any MS signal. But we have found conditions that are compatible with mass spectrometry, and these conditions make use of volatile acids and organic solvents. Now, the lower UV chromatogram here again shows a separation of Herceptin, but this time using a mobile phase composed of 20% acetonitrile, 0% trifluoric acetic acid, and 0.1% formic acid. Now, under these conditions, we can nicely spray our proteins in the mass spectrometer without contaminating our source, and moreover, we do get a very nice mass spectrometric signal. Now, we have to be honest here, our peak is also broader compared to the upper chromatogram. And this is then the downside when going to MS-compatible conditions, you do lose some resolution. Now, as shown in this slide, the size exclusion chromatographic method using acetonitrile, TFA, and formic acid has successfully been applied on intact monoclonal antibodies and also fragments thereof which originate, for example, from reduction, which gives rise to the light chain and the heavy chain, papain digestion, which gives rise to the FAP and the FC fragments, or IDS digestion, which cleaves the monoclonal antibody just below the hinge region. Now, in the detailed characterization of monoclonal antibodies, it is highly advised, and it is even common practice nowadays, to perform measurements at all these levels. Now, measurement of the peptides is of course also very important, but in these cases one will typically not use size exclusion chromatography, but rather reverse phase liquid chromatography. Now, one more thing I would like to say here on this slide is that it is not always evident to analyze the light chain and the heavy chain on size exclusion chromatography. Using a phosphate buffer, for example, the separation shown here of the light chain and the heavy chain cannot be obtained. Now, while we are using an MS-compatible buffer, the chromatograms I have shown you up to now are in fact UV chromatograms. Now, of course, next to the UV data, we simultaneously acquire MS data, and as shown in this slide, UV and MS chromatograms are really very similar in terms of peak widths. Now, shown are on top the overlaid UV chromatograms of intact and papain-digested Herceptin, and at the bottom the MS trace. So it is clear that the path from the UV detector to the MS system does not really contribute substantially to peak dispersion. Now, peak intensities are of course different, which is explained by the different mode of detection. So it is more difficult to ionize intact maps compared to the smaller FAP and FC fragments, and this explains the lower intensity we see for the intact map in the MS trace. Now, this slide also provides some more detail on the size exclusion mass spectrometric conditions. So we made use of the BioSEC column with an internal diameter of 7.8 mm, a column length of 30 cm, and this column was packed with 3 micron particles. Now, in terms of instrumentation, we use an Agilent 1290 Infinity UHPLC, which was combined with an Agilent 6540 accurate mass and high-resolution Q12MS system. Now, the mobile phase composition has already been discussed, so it contains acetonitrile, TFA, and formic acid, and this was delivered to the column at a flow rate of 1 milliliter per minute. Now, you might be surprised by the high flow rate here that we are using, but the easy sources nowadays can easily handle or tolerate these high flow rates. Now, it might as well be interesting here to use a 4.6 mm internal diameter column, since the flow going to the MS can then substantially be reduced, which might give rise to higher sensitivity. Now, in the next couple of slides, I will give you some more detail on the analyses of intact and fragmented maps, this to illustrate the performance of the size exclusion MS method. Now, here again we have the analyses of papain-digested Herceptin. Now, on top you see the MS trace, and at the bottom you see the UV trace. So 5 peaks have been annotated here, and the deconvoluted MS data associated with these peaks are shown in the next couple of slides. So let us first consider the 2 main peaks, which are annotated as A and B. Now, here you see the associated MS data. So note that this is really deconvoluted data and not raw data. So we are really looking here at molecular weight values and not at mass over charge values. Now, the peak A corresponds to the FC region of the monoclonal antibody, and this region is responsible for the effector function of MAPS. Now, several glycoforms can be revealed there that contain the typical mammalian complex type of N-glycans, which are termed G0, G0F, G1F, and G2F. So these structures are displayed there as insets in the spectrum. Now, having a technology capable of measuring glycosylation is extremely important for monoclonal antibodies, since this really represents a critical quality attribute. Now, the peak B that we observe, well, this peak corresponds to the FAP region, and this is the antigen binding part of the monoclonal antibody. Now, both the FC region and the FAP region, well, the measured molecular weight values that we observe there are really below 0.05% of the theoretical molecular weight values, and this is of course expected when using high resolution and accurate mass instrumentation. Now, very important to mention here as well is that we do get some very clean MS data. Now, we are making use of a dedicated size exclusion chromatography column, and this means that apart from the QC testing done at the Agilent factory, this column has not seen any salt. Now, in the past we have tried to perform size exclusion MS measurements on a column that had seen a lot of salt, and the MS data really looked very noisy, and we did observe a lot of sodium adducts. So this is an advice that I can give you here. Please use a dedicated column for size exclusion mass spectrometric experiments. Now, based on the molecular weight measurement, it could further be revealed that the smaller peaks C, D, and E that we observed, well, these correspond to parts of the heavy chain as well as to the light chain. And very important to mention here is that all these fragments have a molecular weight around 25,000 Dalton. And under pure size exclusion chromatographic conditions, these peaks are not expected to be separated. And therefore, under the MS-compatible condition that we are using here, separation is not purely driven by size. On this slide, I'm showing you the size exclusion MS data of intact Herceptin. So on top you see the MS data, and at the bottom you see the UV data. And again, several peaks are annotated here. And as shown on this slide, the main peak A corresponds to the intact antibody with a molecular weight of around 150,000 Dalton. So it is quite remarkable that proteins with molecular weight values that high can successfully be measured, and on top even the glycosylation pattern can be revealed. Now from the MS data, it can further be concluded that the smaller peak, really the trace peaks, which are annotated as B, C, and D, while these correspond to fragments of that monoclonal antibody, which are likely generated during storage. So we do observe partially reduced map there, containing only the heavy chain and the light chain, together with the glycosylated variants. We also see two light chains connected together, and we also see a cysteine-related light chain. Now the last size exclusion MS example I want to show you is related to the measurement of the supernatant of a map-producing show clone and the protein A purified sample. Now, as you all know, protein A binds to the Fc region of monoclonal antibodies, and it can be used to selectively enrich them out of complex protein-containing samples, such as show cell supernatants. Now, this figure here shows a size exclusion MS analysis of the supernatant, and several protein signals are clearly observed here. So the red trace I'm adding now shows a protein A purified sample, and a single peak is observed which elutes at 6.5 minutes, where intact monoclonal antibodies are really expected to elute. Now the early and the late eluting peaks are clearly gone in the protein A purified sample, which means that purification was successful. Now the early eluting peaks at 5.2 minutes in the supernatant clearly corresponds to aggregated material, and based on the elution of the other peaks at 8 and 9.2 minutes, one would annotate these as heavy chain and light chain. So this would mean that a substantial amount of light chain and heavy chain have not formed a functional map inside the show cell. Now to verify the identity, the purified map was reduced and the light chain and the heavy chain measured on size exclusion chromatography. And this is the blue trace that appears. So the light chain and the heavy chain, they do not overlay with the peaks in the supernatant. And this is clearly a situation where mass spectrometry comes into the picture, and using the MS data, all the peaks could nicely be identified. And the identifications are really shown here on top of these peaks. Now, when having a more detailed look in the size exclusion MS measurement of the reduced sample, it is observed that we only obtained partial reduction. So we do still see some intact monoclonal antibody, heavy chain only antibody, and then antibody with one heavy chain and one light chain. And it's not a big surprise that we only obtained partial reduction, since we performed the reduction in a plain buffer without adding any detergents or denaturating compounds. Nevertheless, we can obtain some very interesting information on the map structure. And this slide here shows the spectra of the light and the heavy chain, which correspond to the abundant peaks C and D in the chromatogram. Now, when looking at the heavy chain peak, we again observe different glycoforms. In this case, mainly G0F and G1F. But on top of that, we also observe a peak labeled with the asterisks, and this shows a mass increase of 128 Dalton compared to the main peak. And this can be explained by the presence of a lysine at the C-terminus of the heavy chain. Now, to provide some more background on this particular event, while the heavy chain is cloned with a lysine residue at the C-terminus, and during protein maturation, this lysine is removed by host cell carboxypeptidases. And apparently this process only partially removed the lysine in this particular case. So 10% of the lysine is still left at the C-terminus. When looking at the spectrum of the light chain, so this is the lower spectrum, a signal is observed with a mass increase of 162 Dalton there. And this can be explained by the presence of a hexose at the light chain. And this potentially originates from a glycation event, which is the chemical addition of glucose, which is used in buffers, to the amine groups in the protein, for example the lysine. And this once more illustrates that a lot of information on the characteristics of a protein can be obtained using the described size exclusion MS method. And with that statement, I would like to come to the end of the presentation. So to conclude, we hope that we have been able to show you the importance of size exclusion chromatography in analyzing aggregation and fragmentation, which are really critical quality attributes of biotherapeutics such as monoclonal antibodies. And while the size exclusion UV methods provide some very valuable information on the amount of monomer, dimer, and also aggregates, additional detectors such as mass spectrometry and light scattering do provide some further detail. Now, we have mainly talked about the detectors, but evidently you need a good upfront SEC column, and this we clearly found in the Agilent Bio-SEC3 column, which provides both high resolution and fast separations. And having said that, we would like to thank you for your attention.
Excellent, Koen. Thank you very much. You provided us with some detailed information about the sensitivity and usefulness of mass spec coupled size exclusion chromatography, so we thank you for that. Before we start the Q&A session, I want to let everyone know this is your final chance to submit your questions for our speakers, so hurry up and send them in now. All right, it looks like we have a bunch of good questions already, so let's begin the Q&A. We'll try to get to as many questions as we can. All right, everybody, so let's get to our questions. The first question is going to be for Maureen. And Maureen, is there a need to use an inert LC system to perform SEC measurements?
Sure, let me go ahead and answer that one. And I'm sure Sonia will have some opinions on that. The answer is yes and no. I was going to say some of it depends on your molecules. I personally like to have it around because we've seen differences depending upon the particular antibodies that we're using. Is it an absolute requirement? No, but it's worth making a check. Sonia, what are your thoughts on that?
So it is question 1, isn't it?
It's the question about using the bioinert SEC instrument.
Well, it depends really on especially on the buffer. So when you have a very salty buffer, then it's really recommended to use a bioinert system just to prevent that aggressive salt will damage your system. But it's also depending on the sample used. So when you have samples which are prone to stick to some metals, especially to iron, then you're better with a bioinert system. So I think these 2 points should be considered.
Okay, great. Thanks, guys. So, Sonia, the next question is actually directed towards you. One of our audience members would like to know, what is the wavelength of the laser? The wavelength of the laser, it's
infrared laser, and it is 658 nanometer.
All right, thank you very much for that. Koen, our next question is for you. One of our audience members asked, what is the operating pressure of the 3 micrometer BioSec column?
So the column that we used was the 7.8 millimeter internal diameter column. So we operated it at a flow rate of 1 milliliter per minute, and our back pressure was around 120 bar.
All right, thank you. So Maureen, back to you. We have a question that asks, can you share some figures of merit on the BioSec 3 column, i.e. retention time, area precision, lifetime, molecular weight range, etc.?
Sure. Well, let's deal first with the molecular weight range. We used the 300 angstrom pore size, so the molecular weight range was from 5,000 up to about 1.2 million. So a very reasonable, a good molecular weight range for the proteins that you saw there. In terms of area and retention time, reproducibility, most of the time we are under 2% for a good, reliable system. Lifetime of the column depends, on how much salt and how high a pressure it goes up to, but you should be getting around 500 injections off of that column. For the basic flow rate type information on the 7.8 that Koen and Sonia used in the work, they can go up to over 1 mil a minute and can go up to a maximum pressure of about 240 bar. pH range in general is about 2 to 8.5. Most people aren't going to go up to 8.5. And if you're looking for other more really specific information on the column, just go to the Agilent website and you'll find a lot of the additional specifications and examples of it in use with other monoclonal antibodies.
All right, thanks, Maureen. That was a very detailed answer, so hopefully our audience member was happy with that one. I was. So our next question is going to be for Sonia. Sonia, is the light scattering detector in the multi-detector module multi-angle, or is it set at 90 degrees only?
It is dual angle, so we have 90 degrees and 15.
All right, thank you. So, Koen, we have a question for you. Question asks: in case one uses salt buffers with size exclusion columns, how can peaks be analyzed with mass spectrometry?
Yeah, so in this situation, it is really advised to collect the peak of interest, and then of course you have to desalt them and analyze them on mass spectrometry. So this is more labor intensive compared to the online coupling that we just presented, but this can nowadays also largely be automated. So what we typically do after the peak collection, we perform desalting online, and for that we use a small reverse phase cartridges. So we load our peaks on the reverse phase cartridges, so then the proteins are bound and the salts are really flowing through the column. And after the salts are washed off, the reverse phase cartridge can be placed in line with the MS system, and then you can nicely elute your proteins in the mass spectrometer without having any salts interference. And in that way you can nicely get some very decent spectra.
All right, thanks, Koen. Maureen, back to you again. We have a very interesting question here. The audience member asks, arginine is sometimes used as SEC mobile phase buffer to reduce the peak tailing, but arginine can also increase protein solubility and prevent aggregation. So arginine may give false cleaner result. Is it acceptable to be used for release anyway, or release assay? Sorry.
I was going to say, that is really an interesting question, as you said, Jeff, because you're right. Arginine is used to reduce peak tailing. If the focus is on peak tailing, we usually try and find out if there's something else that's in the tail, so you're not covering up any real result? Is it a low molecular weight component that's in the tail? So try and answer that question first using like the SEC-MS. The other way that arginine gets used is to improve the recovery of the aggregates. So you mentioned the aggregation part of it as well in the question. In most of the cases, in everything I've come across, it does not give you a, I wouldn't call it a false cleaner result. It just gives you a more accurate result of the aggregates. If you're going to use it in a release assay, you're going to need to do just some additional background characterization to prove that you're going to need one of those secondary techniques, whether it be a light scattering or something else that really shows that it's not changing your results, just making them correct, and you'll have to use it consistently in your method.
All right, great. Thank you, Maureen. Sonia, we have another question for you. How does light scattering do with very small peptides as low as dipeptides?
Well, the problem is that light scattering is really, really sensitive to huge molecules like the aggregations from monoclonal antibodies or other proteins. But when you go to very small proteins, and especially when you go to peptides, which are very, very small molecules, the sensitivity is not so good. So if you want to analyze them, you have to inject really a huge amount of it to see something.
Okay, thank you. Moon, we have another question for you. The audience member asks, why is it important to analyze a monoclonal antibody at different levels, i.e. intact, light chain, heavy chain, etc.?
Yeah, so when you work at different levels, you do get some complementary information. Now, for example, when you measure an intact monoclonal antibody, you can verify the molecular weight, and you can also obtain some information on the glycosylation. But when you work at the fragment level, for example, at the light chain of the heavy chain, you start to see some more modification, and like the example we showed you with the glycation and also with the C-terminal lysine. And you can also nicely pinpoint these modifications to specific domains of the antibody. So for example, we could pinpoint the glycation to the light chain, we could pinpoint the lysine presence to the heavy chain. Also the glycosylation, we could pinpoint it to the heavy chain. And this is of course very important. And you can also take this one step further and start to analyze the peptides, because then you can really look at modification at specific amino acids. But then of course, you're no longer looking at size exclusion MS, but then you would rather go for reverse phase liquid chromatography in combination with mass spectrometry.
All right, great. And it looks like we have one final question for Koen as well. Question asks, what is the mass difference one typically observes between the measured protein molecular weight and the theoretical protein molecular weight in SEC-MS experiments?
Yeah, so we made use of a QTOF system, and the mass difference that we observed there between the measured and the theoretical mass is well below 0.005%. So the light chain, for example, it has a molecular weight of around 24 kilodalton. So normally we can measure that within 0.2 Dalton accuracy. And an intact monoclonal antibody, which has a molecular weight of around 150,000 Dalton, we can normally measure that with a deviation of 1 or a couple of Daltons.
All right, thank you very much. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it, or you can watch the whole thing over again, or feel free to forward it to your friends or colleagues, which we always recommend. I'd like to thank Maureen, Sonia, and Koen again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Agilent for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.